My name is Shengliang Ni from the University of Tokyo. It's a great opportunity to present my research here. So as you see, my research is about the repeat elements enriched in the cis-regulatory regions in cancer cell transition. And we guess the repeat elements will have some function in the cancer cell transition with the estrogen-independence. So before introduce my research, I would like to say something about the basic cell line information. In our research, we use the cancer cell line named MCF7, and it's quite typical cancer cell line in this field of research. And some typical characteristics, like the MCF7 cell will have the estrogen receptors and the proliferative response to estrogens. And for the therapy, you always use the surgery and radiation chemotherapy, and the very typical one for the breast cancer are the endocrine therapy. Here is a very simple example for the endocrine therapy. First, for example, that's the MCF7 cell. We can perform a very simple estrogen deprivation for the cell, and normally most of the cell will die after the deprivation. But somehow, parts of this kind of MCF7 cell, after long-term culture with about 2 or 3 or 4 months long culture, they will acquire resistance. And this type of cell, we named them as the LTAT. And LTAT, the whole name is the long-term estrogen deprivation cell type, and we call them the endocrine therapy resistant. And actually, the MCF7 cells survive after the whole process. So we are thinking why this happened, and the first thing came to our mind are the regulatory elements. So we do think that it's quite important to understand the mechanism involved the development of resistance and cell type transition, because this could represent one type of the cancer cell. They have the endocrine therapy resistance. So about the regulatory elements, look at this graph. I think that's a possible made very typical model for enhancement promoters and enhances promoters. And we also think that it's, of course, the enhancement promoter may have some function in this process. So we decide to look at some special enhancement promoter. So we first check with the transposable elements relics in the enhancement promoters. And these are some previous research show that the transposable elements and short simple repeats may act as enhancer or have some distribution with the promoters in this previous research. And also the very current research published in 2023 mentioned that the transposable elements will help enhance an upstream promoter RNase interaction by simple base pairing and help them to have the 3D architecture in the chromosome. And, like the model in the very right here. So we start our research from here. So to study the cancer cell transition in these two types of cells, we use the cage-seq technology. Name is a cap analysis for genome expression and deep sequences. It's a simple technology to measure RNA expression and very good to map the transcription start sites. And when we have the cage-seq data, and we will use, because cage-seq data, they add a cap at the very first position after the 5', it could be very nice to map to the transcription start site. So now we have a transcription start site. We use another deep learning software to predict the cis-regulated elements in the data. So we use the ESCAPE to predict the enhancers and promoters based on the transcription start site from cage data. And after that, we use another novel advanced technology named radical-seq. It is a technology, as like the name mentioned, it is a technology for sequences like RNA and DNA interacting complexity the technology, and it's quite good to identify the gene-wide RNA chromatin interaction. It's quite complicated technology, so I won't show too many details like in this technology. And all the things about this small structure, and we capture the RNA and DNA like this, and make some knock out the protein and the DNA, part of the DNA, and sequences the RNA and the DNA here. And then we can got to the coordinate and map to the reference genome to got the RNA and DNA coordinates. So there are some points. We use KH-seq to get the transcription start site, and use ESCAPE to get the cis-regulatory elements, and like this. And we have the repeat marker for repeat elements annotation to find the repeat elements containing enhancers and promoters, and like this. And that two enhancers and promoters are the repeat element containing. And after that, we use radical-seq to find the repeat elements containing proximity link, because radical-seq will find the RNA and DNA interactions in the genome. Like, so we got the promoter enhancer cis interaction and the trans interaction. And of course, we have a EE cis trans interaction and PP cis trans interactions here. And because the radical-seq will find an RNA and chromatin interaction, so that the case in our research are like this: e-RNA and the promoted chromatin, or maybe the p-RNA, promoter RNA with the enhancer DNA chromatin here. So that's the case in our research. But until now, we only analyze the promoter enhancer cis interactions, and of course, we combined both of the case together to simply understand. But maybe we can have a deeper analysis here. Okay, so we have the very first look at our data. So first we look at the percentage of cis-regulated elements in MCF7 and LTAT cells. Look how the regulated elements intersect with the repeat elements. And you can see the LTAT, the enhancer were annotated with the blue bars, and the promoters were annotated with the green bars. We can see that enhancers are more like to, and of course the repeat elements average percentage in human genome were annotated with the red bars. And we also find that in the enhancer more like to, more like to intersect with different types of the transposable elements, like, most are the LI1, ALU, and MIR, and of course the types of LTR. But surprisingly, we find that a very typical LTR transposable elements like ERV1, ERVK, they don't have any intersection with our regulated elements here. But I annotated red square here because we find something new. And next we look at the unique upregulated elements in LTAT cells. We're comparing with the MCF7, and they are all of the upregulated elements in LTAT, and we find that as the same, we have this enhancer in the blue bars and the promoters in gray bars, and the average percentage of repeat elements in red bars. And we find that they have the same tendency with the previous slide, and also find that the ERV1 repeat elements have a significantly increased considering the upregulated regular elements in LTAT cells. So we think that's quite consistent with our hypothesis. So we think maybe the repeat element have some function in these both types of the cell type, in both of the types of the cancer cells. So we did the deep analysis for this. Yeah, we did the deep analysis here, but it's also quite simple. We count how many links we found in the radical-seq to as a measure for how strong the interaction of these both regions of RNA and DNA. So we count the links based on the radical-seq. We calculate the count fold change in LTAT and MCF7, and those with blue one are the interaction they higher in MCF7, and those with the red one are the interaction they have a much higher count in LTAT. And for example, each dot, for example, this one, the top one, represent the simple repeats in promoter, the DNA HAT transposable elements in enhancer the types of these kind of interactions. And interestingly, we find that most of the interaction, they have a large amount, they are higher in MCF7, they are related, the enhancer interaction are always related with the DNA transposable elements. And of course, we find the top second are the LTR transposable elements in the enhancer. And of course, we do the same thing. We think it's quite interesting, so we do the same thing. But this time we consider all of the chromosome and regulated elements and all of the repeat elements. For example, these dots means the SI and the MIR repeat transposable elements intersect with the promoter in the chromosome 20. And we found a lot of the, how to say that, they're significantly high or significantly decreased interaction here. And most interesting is these two tops of those. They're significantly increased and decreased interaction. And fortunately, they coinciding with some interesting gene like NCOE3. They are the endocrine therapy resistant gene, and of course the MALAT1. MALAT1 is also a very famous, how to say that, no long coding RNA in the cancer cells. So we do think this could be do more analysis for this for the repeat elements in both cancer cells. So we have some typical examples here, like we choose one ESR1 receptor, and we find that in LTAT and in MCF7 that the linear chromosome distance of this type of the interaction related to the breast cancer related gene ESR1, they're quite different. For example, they're extremely long in LTAT, but not in MCF7. And of course, we are thinking like if the repeat elements have no function with the interaction, so we also check with the interaction with the repeat elements or without repeat elements. And we can see that the interaction with repeat elements are much significantly longer than the interaction without repeat elements. Another example are the gene, namely ERBB4, always co-repress with ESR1, and it's quite interesting because we find there is an extremely hot spot for the interaction in the ERBB4 locus, but we cannot find any interaction in the same locus in the MCF7 here. At very first, and we think that's because the expression level of the ERBB4 increased a lot, so that makes the mRNA for the ERBB4 everywhere, and that's why we can find the ERBB4 can map to the every coordinates in the chromosome 2. But our partner and our collaborator helped us to perform some experiment. They find that the expression level, they have some increase, but not that much. So we think that's not because of expression level. So yeah, and we also do the same thing with the interaction in LTAT and with the repeat elements or without repeat element. We find the same thing, that the repeat elements containing interaction are much longer than the interaction without repeat elements. So of course, we also calculate how the interaction distance in the whole genome and find that about there are some chromosomes, they have more long-range repeats containing interaction observed, and of course there are some less long-range repeat containing interaction observed in this kind of chromosome. Yeah, and also we think it's interesting to study the genes that might have interaction in these chromosomes. And I'm sorry to say that I have to make some summary here because this research is ongoing. We didn't finish we don't have a very clear conclusion for our finding. So to summarize our research, like we find that in this LTAT MCF7 transition, the whole genome-wide pairing of repeats in enhancer and promoter has significantly changed, especially the simple repeats to DNA TEs and the LTR TEs. And some interaction around breast cancer-related genes containing repeats are more inclined to be long distance. And and also we find that very interaction enriched both at the ERBB4 locus in LTAT cells. And we plan to analyze all of the cis interaction, trans interaction, and use the high C data and the TAT regions. But until now, we don't have a very accurate high C data because they use a different enzyme for the LTAT and MCF7. And of course, we just finished the ChIP-seq data, so we'll use this for validate some high significant enhancers promoters. And of course, in future, we are planning to validate the transcribed repeat sequences. And of course, the system analysis of a breast cancer-related gene is very necessary. And finally, I would like to thank our collaborator. They are the Noriko Saito sensei from the Cancer Institute and GFCR, and Masaki Kato and Piero Konichi professor. They are from the RIKEN IMS Center because they providing the samples and the data for the sequences and very helpful suggestions for this research. And all of this research and data and samples are all unpublished. So I will especially thank all of them and also thank the members of our lab in the University of Tokyo. And that's all. Thank you.
Well, thank you, Shengliang, for the very interesting and very exciting talk. Oh, we have questions from the floor already. Go ahead.
Hi, thank you for the very interesting talk. I had two questions, but then I saw your future plan and one already answered, so I had to skip that one. I'm curious about whether you have given some thought when you detect these long-range interactions. So I do a lot of research in TF-TF interactions, and one of the things we're interested in is these long-range mediated chromatin interactions. So you have, like in your initial drawing, you have the chromosome, right?
Yeah.
If you go back at the very beginning, perhaps that would be...
Very beginning, I mean, you mean the introduction part?
Yes, where you draw the long-range interaction. Yeah, I know I'm making you work hard, I'm sorry.
This one, or?
Slightly more, maybe? Yeah, that one.
Okay, I see.
Perfect. So I was thinking, have you tried to look into two things? One, whether these long-range interacting, you did it for, I think you did it for the estrogen receptor and ERBB4, but have you looked whether there are other motifs that are enriched?
Yeah.
Maybe using some SIA or other simple tools?
Give me a chance to make the slides. Yeah. Actually, I do check the motif for the region for the radical-seq font, like the RNA and DNA region. It's quite easy to find. They have some very convincing or very high possibility motifs. But when I do the same thing with the random enhancement promoter, they can find the same thing. So we do care thinking about the motif thing, but maybe not the only one evidence for that. But we will check for that. And another thing is we use a very simple motif tools like meme suites, but how to say that? We check maybe I didn't find, maybe I'm not very expert for the meme suites because we find that motifs are quite short. So we're thinking to perform the very long time multiple sequence alignment to find the consistent sequences for the regions that we found. Maybe that can map. We can see them as a motif to find if it's that possible for them. And just like the research I mentioned here, this research, they do the multiple sequence alignment to find the motif because they find that allele elements in the enhancement promoter in 6 types of cell line, they find quite long base pair, quite long motifs for with the multiple sequence alignment. So I guess I will try with this, but thank you for the advice.
I have just a lightning quick follow-up, really quick.
Sorry, I think we're running out of time already. Perhaps, right, Alvin, if you have a quick one.
Okay. No, great talk. I really enjoyed it. Is there any difference in terms of the line 1 expression between the two MCF7, is it, and LTAT?
You mean the difference between MCF7 and LTAT?
Yes, because I think one of the interesting things is to compare whether there are any somatic changes in repeat elements and to see whether there's any of the changes you see between the 2 time points.
You mean the experimental level difference or the...
So essentially the change, the differences in the enhancers that were
they do have some I guess you're asking the difference between this MCF7 and LTAT.
Yeah.
They do have some difference. For example, like the MCF, we know the MCF7 are the estrogen receptors positive type cell line, but then we find that the LTAT became like, how to say that? I'm not an expert in the MCF and these two types of cells because the data are from our collaborator. But from my understanding in the cancer research, we find that for example, the estrogen receptor gene, the ESR1 gene, is extremely high expressed in LTAT, and they have some super enhances region in the ESR1. But the knowledge for the LTAT are still not that much, so we're still keeping studying there. Yeah, especially like the regulatory network, we don't know that about.
Okay, thank you so
Sorry.
much.